One of the big things we did in Food4Me was to test the hypothesis that personalised nutrition would work. And the next presentation is going to give you a summary of those findings. And I think in advance, I can say that it has taken us longer to get through the project than we thought. And therefore, we're not as advanced as we are in analysing all the data. So probably in the questions, you'll have many questions to ask, some of which we will not be able to answer yet. But John is going to give you a high-level analysis of what has been observed in the Food4Me project.
Mike, thank you very much for the introduction, and it's great to be here to tell you about the proof of principle study. So this was a study where we set out to test the hypothesis that personalised nutrition would do better than we've been able to do up to now using conventional approaches. So we already had the case made that what we eat really matters in terms of our health and well-being. And if you look across the whole range of public health, almost every issue that you face in modern society is influenced by food choices. It's of course most obvious in the huge increase in obesity and in conditions related to obesity, which has happened within one generation. The explosion in obesity worldwide has been a phenomenon which has really happened over a very, very short period of time, 30 to 40 years. Quite remarkable. And of course, part of that is related to food. So understanding how we make food choices and understanding how to change people's food choices is important in helping to improve public health. Now, of course, there have been a number of approaches to changing people's food choices in many parts of the world over the last decade or so. And they have had some successes, but relatively modest. And if I could just draw your attention to this one on the bottom of the slide, which refers to attempts to help us to eat more fruit and vegetables. We all know that we should eat at least 5 portions of fruit and veg a day. And what this yellow line shows is the change in the proportion of people who are eating 5 portions of fruit and veg per day over about a 10-year period. And you see it initially, it began to climb, but then it's fallen back and plateaued. So we made a little bit of progress, but not a lot. And less than a quarter of us are managed to eat 5 portions of fruit and veg a day. So there's plenty of room for improvement. So we asked the question, and this was a core part of the work that we did within Food4Me. Would it be better, would we do better in terms of improving people's food choices if we move from the current approach, which is essentially a one-size-fits-all approach, to a more person-centered approach, where we tried to make the intervention relevant to the individual? So would personalised nutrition do better? And in this area, it's all about helping people to change behaviour, changing their food choice behaviour and doing that in a sustained way. There's huge interest in this area, and Mike in his introduction referred to all the things which have come from the sequencing of the human genome. And I guess one of the most recent manifestations of that has been Mr. Obama's push and contribution of additional dollars to the NIH for what he calls precision medicine. But basically, it's the same idea repackaged, that if we could design interventions which are relevant to the individual, we would do better. Now we know, and we have a growing body of evidence about these links between nutrition, between our dietary choices and health. And the evidence base for that is increasing month by month. And the idea is that if we had appropriate genetic information, this might help to identify the right diet for you and the right diet for me. And in addition, it might motivate us to adopt healthier eating patterns. So we might use that information to help with choices and also to sustain those choices in the long run. As Hanny said, the developments in technology have been dramatic. When the human genome was first sequenced It took about 10 years, about $100 million. Now you can sequence a human genome for about $1,000 in a day. It's astonishing. The quality of the evidence is better, it's much faster, and of course it's very, very much cheaper. So it no longer becomes an impediment to know about the genome. We have that kind of information. The question is, can we use it? Now, when we set up Food4Me, there were lots of papers around saying that this is potentially a good idea. But there actually were very few studies which attempted to test the idea. And just about the time we started, there was a systematic review as part of the Cochrane exercise to examine the evidence that providing DNA-based advice would help people to change behavior. And the authors of that report, this is 2010, said claims that receiving DNA-based test results motivate people to change behavior are not supported by the evidence. And the authors of this review, a very authoritative review, made the call for larger and better quality randomized controlled trials. So just as in other areas of public health or biomedicine, we need trials to demonstrate whether or not something new works. So that was the background to the proof of principle study that we carried out as part of Food4Me. So this was a trial of the idea that personalizing nutrition advice might help us to do better. So our hypothesis was that if we provided appropriate personalized dietary advice, it would improve our dietary intakes and a range of markers of health, including our body weight and waist circumference. And we took that hypothesis and we addressed a number of research questions. In these kinds of studies, you need to compare your new treatment or your new approach with a standard approach. So we said, is personalized nutrition advice more effective than general health eating guidelines? So is what we're proposing better than the standard model today? And then we took what we're learning about the new science, about phenotype and about genotype, and asked whether including information about phenotype and genotype would be more effective than just knowing about people's diet. We didn't know how much information to give people, so we said, would more feedback improve compliance? And because we were trying to test what might work in real life, we chose to deliver our intervention via the internet. And we think that in the long run, if these sorts of interventions are going to make a difference to public health, digital technologies will be critical to making them work. So we designed a randomized controlled trial. It involved European adults. We attempted to recruit quite a large number of individuals. Over 1,500 people was our target. And those people were going to be randomized into 4 treatment groups so we could make direct comparisons. We would contact these people and work with them via the internet, and we would look for changes over a reasonable length of time. We would look for changes sustained over 6 months. So we set out to recruit people in various parts of Europe, and we recruited in 7 different European countries and had a target of recruiting 220 people in each of those 7 countries. We used the Food4Me website to attract individuals to this study, and the website then became the portal through which they communicated with us. So the individual would learn about the study, perhaps through a press release or a newspaper article or an advertisement, and then go to the website and join the study. And that then became the way in which we carried out all the procedures which are critical for running a trial. And I want to emphasize that we ran this trial using the most robust procedures, the sorts of procedures which would pass muster with any high-quality trial. So we had the usual things about collecting information about the individual, and going through a process of consenting. Because we are going to ask people to change their diet, we needed to exclude a few people who might have some disadvantages from changing of the diet. So, for example, we didn't recruit people who were pregnant or lactating or had food allergies or intolerances. But by and large, then we recruited all European adults. And those people then were randomized to 4 treatments. And they're shown here in these 4 boxes. So level 1 is our control group. And these are the individuals who got the standard package, the one-size-fits-all conventional approach to changing diet. So this is the best that current things could tell us. And then the other 3 levels, 1, 2, and 3, represented different ways of personalizing nutrition advice. So level 1 personalization was based on an analysis of the individual's current diet, so helping them to change what they're currently doing towards something which would be healthier. In level 2, we did that, but we also analyzed their current phenotype. So based on information about their body size, their body shape, and constituents in their blood, we then tailored the dietary advice. In level 3, we did both of those, diet and phenotype, but we also analyzed their genome and provided advice based on their individual genetic variants. So this allowed us then to compare the standard control, one-size-fits-all intervention, with 3 types of personalization. And as we go through and I show you the results, we'll look at it in that way. In all studies, you need to be very clear what you're trying to measure. And so we have sets of outcomes, primary and secondary outcomes, and also interim outcomes. So our primary outcome, the thing we were most interested in, would be the dietary intake of these individuals 6 months after they've been part of our study. So after 6 months, had they changed their diet, and was it a change in the right direction? So are they now eating a healthier diet, a diet which we think would be better for them in the long run? We, of course, measured lots of other things. We measured their physical activity. We measured a whole range of phenotypic biomarkers. We measured those throughout the study, but we're particularly interested in the long-term effects up to 6 months. And then we had some interim outcomes at 3 months for all of those factors. Because of the shortage of time this morning, I'm going to focus on the primary outcomes. But of course, we have a wealth of data on the secondary outcomes and the interim outcomes. So let me say a little bit more about the people who were in the control group. So these were the people who were getting generic, one-size-fits-all dietary advice. So that involved information encouraging them to eat more fruit and vegetables, 5 portions of fruit and veg per day, to eat more whole grains, oily fish, low-fat dairy products, reduce intake of red and processed meat, to alter their type of fat towards a more healthy pattern of fatty acids, and to reduce salt intake. So these are all the standard things that we all know about, and health authorities around the world have been trying to change people's diet in this way. In addition, if people were overweight or obese, we gave them advice on total energy intake to help them to reduce their body weight. So now some information about the personalized nutrition advice. So this is levels 1 to 3. So we delivered this personalized nutrition advice digitally, and they were delivered based on the participants' baseline report and what they told us they were doing at month 3. We identified for everyone 3 food-based goals. So the reason for focusing on a food-based goal is that we eat foods, we don't eat nutrients. So telling people that they should eat more vitamin C is not very helpful, but if you tell them that oranges are a good source of vitamin C, they know what to do. We chose those goals by identifying what was most important to change in the individual's diet. So we ranked the risk of poor diet or adverse phenotype or their genotypic risk markers. We ranked them and we chose the top ones. And we help them to see that by using one of these little bars, a green, amber, and red. And we can place the individual on the bar to show where they currently were. So this is an example for salt, and this person is in the red bar, they're eating too much salt. Okay. We needed information on what they were currently doing. So to do that, we need to collect detailed dietary information. And so we developed and validated, as part of Food4Me, an online food frequency questionnaire. So individuals could use the website to tell us how frequently they consumed all of these foods. Now, frequency is good, but if you're going to do quantitative studies, you have to also ask the how much question, not just what did you eat, but how much did you eat. And so our food frequency questionnaire included an attempt to quantify that by providing pictures of portion sizes, so the individual could choose which portion size was appropriate for them, and also provide information on the type of food, so not just that they ate bread, but it was this one, not that one. Doing studies in 7 European countries is a tough challenge. Doing it when you don't meet up with the participants is an even tougher challenge, because we needed information on their phenotype and we needed information on their genotype, and they were out there somewhere and we were in our universities. So that meant we had to collect all this information remotely. So we asked the participants to do it for us. Hani had talked about the kind of developments which are out there now to help people do this. So we trialed that approach within our study. So we asked people to measure and record their height, their weight, their body measurements. To help them to do that, we provided information in video format and in written form in the languages of the 7 countries so that they knew how to do this in a standardized way. We also want to measure things on their blood. So for that, we needed blood. So how do you get blood from out there into the university without having a nurse go out and collect it? So what we said, okay, collect it yourselves. So this is using these little dried blood spot cards. So the individual pricks their finger, you get a little blood drop, you touch it on the filter paper, and it collects a little blood spot like this. Many of our colleagues were very skeptical. People will never do it. It's too painful. It's not a nice thing to do. But they were wrong. The wonderful people out there in those 7 countries did it, and they did it brilliantly. So they filled 2 of these cards, 5 blood spots on each card, and with those, they then sent them back to us by post, and we measured a whole range of nutritional and metabolic markers. We had a similar issue with DNA. We needed some DNA, and so we asked people to collect a buccal swab like this at home, pop it in the post and send it back to us. And using the DNA extracted from the cells in those buccal swabs, we measured 32 nutrient-related genetic variants. And we used 5 of those variants to tailor the dietary advice offered within the study. Again, we had instructions for all of the measurements, instructions for all of the measurements in video and printed formats. And the participants did brilliantly. 95% of them managed to collect their blood, which is far, far better than I ever imagined. They also collect information on their physical activity by wearing a little monitor, and 98% of them collected the buccal swabs for their DNA measurements. So compliance was fantastic. So on the DNA side, we had to select some genetic variants to test, to examine the proof of principle that this kind of information would be helpful to people in making appropriate dietary changes. And so an important part of the work was to examine the scientific literature to identify those genetic variants for which there was the best evidence of a link between diet, the genetic variant, and health outcomes. And so we selected these 5 genes, a gene which is involved in the metabolism of the B vitamin folate. And if you have a particular variant of that gene, then you need more folate. So it's a way of encouraging people to consume more folate. The FTO gene is involved in how big we are, how fat we are. So if you have the risk variant for FTO, then to maintain a healthy body weight, you need to reduce your food energy intake or take more physical activity. We had another one, TCF7L2, where you get better weight loss on a low-fat diet. So if you have that variant, then a lower fat diet would be good for you. Mike's already referred to ApoE, and ApoE4 is the variant which is associated with higher risk of cardiovascular disease, heart disease, and stroke. It's also associated with higher risk of dementia. And we know that if you want to maintain healthy blood cholesterol levels, and you've got ApoE4, you need to lower your saturated fat intake. And finally, a gene which is involved in metabolizing fatty acids, FADS1. And we can benefit, in this case, by consuming more of the omega-3 fatty acids. So these were 5 genetic variants for which we felt the scientific evidence was strongest. The next challenge was putting all that together, because now we have a wealth of information about the individual's diet. We know a lot about their phenotype, and we know quite a lot about their genotype. And we had to make it all fit together. And to do that, we developed algorithms, or decision trees, which allowed us a hierarchy of decisions and eventually ended up with personalized nutrition advice. So in this example, we start with information about the FTO gene. This is the gene associated with body size, how fat we are. So we know whether the individual carries or does not carry the risk variant. We now look at their body weight. So if they carry the risk variant and they're too heavy, we can tailor the advice appropriately. We now look at where they're carrying their fat. Have they got a normal waist circumference, or they're a bit too big around the middle? Next, we look at their physical activity. Then we look at their blood glucose, and eventually we get all of these individual pieces of advice. And so we can tailor the advice for the individual based on all of these pieces of information. And we had over 300 different possible personalized nutritional advice. As I said, we tried to provide feedback to people in ways that would be easy for them to understand. So we used these bars to represent where people currently were. So for a whole range of components of their diet. So were they in the green area, which is fine? Were they in the amber area, where they need to watch out, maybe begin to make changes? Or were in the red area, where they really need to make some changes? And then we gave them detailed information about how to make those changes. We selected those top 3 dietary changes they need to make, and we explained in some detail how they might make those changes. So in the final part of the presentation, I'm going to give you the results of the study. And I'm going to start by addressing the question, who was interested in participating in personalized nutrition intervention? But that's rather important to know in terms of how this might be translated to the wider community. So when we advertised for participants in the study across the 7 countries, over 5,500 people went to the website and joined up. A wonderful response. So we learned a lot from the characteristics of those individuals. Nearly 2/3 of them were female. And that wasn't a surprise, because almost anything to do with health or lifestyle, you get a far better response for women than you do from men. Men seem to be hopeless in this respect. A wide range of adults, from 15 to 87. When we started, we thought it would be, the 20s and 30-year-olds who would join the study. Not a bit of it. People across the whole of the range. And the mean age of those 5,500 people was 40. Some of them were already on a therapeutic diet, nearly 7%. 12% told us that they had a food allergy or a food intolerance. And of course, they were all people who had access to internet because that's how we were communicating with them. We were intrigued to know whether people would be joining the study because they want to lose weight. So would these be all the rather plump people across Europe who thought this was a cheap and easy way of helping them lose weight? But actually it didn't turn out to be like that at all. The people who joined the study were pretty much like the rest of European adults. So if we look at the final bars, which are the means for people in Europe, the grey bar, and the people who joined Food4Me, they're almost identical. So we were selecting, or people who were selecting themselves to join the study who were pretty much like European adults. So those were the 5,500 who signed up saying they would like to join the study. We then selected from that those people who met our criteria to join the study, and they were randomized into the randomized controlled trial. So the next question was, what were the characteristics of the people that we randomized? So we randomized over 1,600 people. So actually, we did better than we expected. We were planning to recruit 1,540 people. We actually randomized over 1,600. Very unusual for a trial. Most trials fail to recruit enough people. We exceeded our recruitment target. Again, more than 50% were women. They covered a wide age range, 18 to 79, the mean age 40. Most of them were white. In terms of adiposity, about 50% were normal weight. This is the green segment of the pie. And we had proportions of overweight and obese people, which were pretty much like the rest of European adults. This shows the variation in body mass index by country, the 7 countries of the study, from Germany at this end to the UK at this end. And the most overweight and obese participants were from Spain, and we had the fewest of those recruits in Germany. So there's some variation among countries, but to some extent that reflects the demographics of the individual country. Now, if you're going to do a robust study, you need to make sure you collect good data. And the innovative aspects of the design of our study meant that we had some big questions to answer. Questions about data integrity. And we identified 2 major issues. The first was, would the measurements that the participants made themselves be reliable? So would they really tell us how heavy they were? Or would they lie about that? And we know that often when people are asked questions, they give answers which reflect a little bit better on themselves. So they're always a little bit taller and a little bit leaner than they really are. Okay? So we need to know whether, in our study, the same would be true. The other issue was about identity. And identity fraud is a potential risk for any internet-related activity. So people might pretend to be someone that they're not. So they might sign up and say that they're a 36-year-old female when they're actually a 47-year-old male. And they might, that would screw the whole thing. So we need to know whether the people were who they claim to be. And to deal with that, we built into the study a subset, a validation study. So 10% of the sample we brought into the research centers around Europe to assess their identity and to repeat the measurements. And we did this very quickly after they made the measurements themselves. So for identity, we looked at them. So does this look like a male or a female? Okay. But we also were a little bit sneaky, and we collected some DNA. We measured the people and to see whether the measurements they had made were similar to the measurements we made. So first of all, the measurements they made and the measurements we made. So this is height that we measured, and this is the height that they reported. And you can see they're pretty good. This is the weight that we measured. This is the weight they reported. Again, pretty good. And the derived variable BMI. So we were pretty confident that we had instructed people appropriately and how to make the measurements at home, and they could do it and do it well. Now, the identity validation was a very novel part. As far as we know, no one has attempted to do this before. So we decided to use genotypic information as our way of determining whether the person really was who they said they were. When each of us was conceived, so at that moment of conception, we acquired from mum and dad a constellation of genetic variants which makes us unique. So unless you're a monozygotic twin, identical twin, your genotypic variance is different from the person sitting next to you. So we had analyzed 32 genetic variants as part of the study. So we repeated that in the validation trial. And what we discovered was more than 99.9% identity between the two measurements. So we're pretty confident that the people who joined the study really were the people who were in the study. So we're coming now to the results of the trial itself. So we had a couple of research questions. I'm reminding you of those. So we want to know whether personalized nutrition advice was more effective than the conventional one-size-fits-all approach. And we want to know what was the best basis for personalization. So is personalization based on phenotype and genotype more effective than personalization based just on an analysis of current diet? Now, in the slides which follows, they're all in a similar format. So I'm going to take you through this one rather slowly so we understand the format of the information, and that will make it easy. So what we're seeing is the change in intake of something, in this case it's energy, at 6 months, so the change from baseline. So what differed after 6 months? The information is provided on a scale on the left-hand side, and we have 5 bars. The bar on the extreme left is the change for the control group. Next to that is the bar for all 3 personalized nutrition groups. And then the 3 gray bars represent each of those groups individually. So this is level 1, the people who are randomized to personalized nutrition based on analysis of their current diet. Level 2, an analysis of diet and phenotype. And level 3, diet, phenotype, and genotype. Now, the first thing I should say is that being part of our study was a good thing for everybody. So whether you were in the control group or in one of the personalized nutrition groups, you did better. So people improved their diets in all groups. So first of all, the comparison between the control group and all of the personalized nutrition groups. So overall, people reported consuming less energy after 6 months in the control group, but even less in the personalized nutrition group. And that's probably appropriate because if you remember, about 50% of the people who are overweight or obese. So overall, we would do with eating a little bit less. Significantly improved change in energy intake in the personalized nutrition group. But then when we look at the 3 different levels, there's no differences. So each of the 3 levels were very similar. So any kind of personalization improved things over the one-size-fits-all in terms of energy intake. So here are similar data for red meat, organized in exactly the same way. So this is the change from baseline. And for most people, reducing their intake of red and processed meat is associated with improvements in health. So what happened? The control group reduced their intake of red meat by about 12 grams per day, but those who were randomized to personal nutrition did better, did significantly better. What about the 3 levels of personalization? Pretty much the same. To try to get an idea of whether overall we were seeing healthy changes, we took all the information for the diets and we calculated what's known as a healthy eating index. So this takes account of several factors of the diet and brings them all together into one index. and a bigger index represents a healthier diet. So these are the information on the change in healthy eating index for those groups after 6 months. Now what you see is what I said at the beginning. Being part of the trial was good for people. So the control group improved their healthy eating index, but those who were randomized to personalized nutrition did significantly better. So a bigger change in their healthy eating index if they were in the personalized nutrition group. But no significant differences between the 3 levels of personalization. So personalization worked, but it didn't matter whether you included phenotypic or genotypic information. So far I've been talking about aspects of the overall diet. But in our advice to people in the personalized nutrition groups, we targeted particular aspects of the diet which needed changing. And across the 7 European countries, these are the top 5 nutrients within the diet which needed to be improved. So number 1 was salt. So across all 1,607 people, on average, most people needed to make a bigger change to salt than anything else. The next most important is saturated fat, then dietary fiber, then folate, and then polyunsaturated fat. So in the next part of the analysis, what I'm going to show you are the data where I restricted through the analysis to those people who received advice for these specific nutrients. So when we gave them advice relevant to these nutrients, could they make appropriate changes? So here are the changes in salt intake, organized in the same way, the control group, the personalized nutrition group. Great news was that the people in the control group reduced their salt intake, reduced it by about 1.4 grams per day, which is a huge and very important improvement. If we could do that across the whole of Europe, we would make an enormous change in the risk of hypertension, and there'd be far fewer strokes and far less cardiovascular disease. The personalized nutrition group did better. They reduced their salt intake by 1.8 grams per day, significantly better. So personalized nutrition worked in reducing salt intake, but no differences between the 3 types of personalization. For saturated fat, again, the control group made improvements. They reduced their saturated fat intake. But the personalized nutrition group did very, very much better, more than twice as well, more than twice as big a change in saturated fat intake. But again, no differences between the 3 types of personalization. And finally, for folate, the control group consumed more folate, but the personalized nutrition group consumed twice as much more than the control group, significantly more. As before, no difference between the 3 groups. We have a huge amount of information on these individuals, and we're just beginning, as Mike said, to dig into the details of that study. So there's a great deal of work still to be done. And we're particularly interested in who benefited from the intervention. Were there particular people who responded better or less well to the intervention? So, for example, did men and women do equally well? What about older people and younger people? Was it really that the plumper people, those people who need to lose a bit of weight, were the ones who were benefiting, and the leaner people not so much? So we need to know about adiposity. What about genotype? We know about 32 genetic variants. Maybe some of the people with particular variants responded better than others. And of course, we want to know about intervention intensity. So giving people more information, recording more things, does that help them to make bigger changes? That's all work in progress. So to summarize the take-home messages, and I think it's pretty clear that personalized nutrition works. We've shown consistently across all the things we've measured that the people who were randomized to personalized nutrition did better than the control group. We saw no added advantages of using phenotypic or genetic information. And we've also demonstrated that doing this via the internet can be effective. This sort of work requires a big team of people. And I want to use my last slide to acknowledge the huge input, first of all, from all the participants, those 5,500 people who went to the website, signed up, and gave us lots of information about themselves. And also 1,607 who joined the study and stayed with us for 6 months. And finally, the teams in all the centres who ran the study. Thank you very much indeed.